spores, I would say, right? Which are useful structures because such a structure I isolated out of the sea. And in the sea, we call those structures the giant viruses because they have a much larger genetic material, or let's better say nucleic acid, than bacteriophages have. And this is what I found in the marine algae. When it's too cold, too hot for this algae, it's not building its real spores, replicating through this form. No, it builds much smaller ones, which are producing energy because nucleic acid is producing energy. This is primary task for nucleic acid, energy production. So, and this is what, so that bacteriophages became the model how viruses after 52 look like, right? And you see it when you go to the Nobel laureate web page, then you'll find inside the justification for the Nobel Prize for the phage group. They received the Nobel Prize in '69. You'll find that the bacteriophages became the basis of all field of virology, right? Wow. And there they are in this easy trap, which we easily can see with the 7 steps. 3 we had already, because they believed when cells are dying, they would transform their material into viral material, like bacteria, certain kind of bacteria are doing when they can't grow anymore because of inbreeding, because of highly intoxication. So this bacteriophage, which does exist, which is similar to the giant viruses, which are called now viroplankton, and it become known that we have such a high amount of this nucleic acid in all seas, so that the organic matter of those so-called giant plankton, it's higher than every organic forms put together. Imagine this. So the ocean, it's full with nucleic acid of giant viruses. But please don't tell, otherwise we could only swim with full body condoms, right?
With a condom, yeah.
Yeah, I mean, nowadays you have to be really cautious, you know. So, but it was your question how if it's definitely sure to say that there is no SARS-CoV-2 outside, and not only SARS-CoV-2, HIV, measles virus, Ebola. So how to see this? Let's go into the 4th step, what virologists are doing. What are they doing? They are isolating nucleic acid, which is breaking down in the decay. So millions of very small pieces of nucleic acid is isolated, is sequenced, and then they read the short sequences, the sequences of the very short pieces, and they add it mentally to a big one, which they never isolated out of a virus and which they never saw.
And this is the in silico piece, right? This is the theoretical, yeah.
And it's easy like this. I mean, since four centuries, nucleic acid easily can be shown to exist when you concentrate it in the process of gel electrophoresis. You see a size separation, and when you have one area of the same size of several thousand molecules of nucleic acid at one point, you can easily stain it and see it. You can easily get it out of the gel and even photograph it in the electron microscope, which I did with the nucleic acid of this giant virus, which I believed in the beginning of my study as a harmless virus, right? Now I call it a mini spore, because it's become known that those particles have their own biochemical activity and that they are growing. They're getting more and more complex. So we have a building block of life misinterpreted as a virus. First bacteriophage, then giant viruses, and virology, poor virology, due to the Nobel Prize of Enders for his polio, piece of brain and chicken fibroblast finding, that viruses are not tissue specific. He got the Nobel Prize for this, for a virology which definitely was overthrown in 1954, but the public didn't know. And the Nobel Prize had the function to keep up with this virology, because they run all the vaccination and would not abandon the vaccination. Even the model fall apart and was disproven. So this paper, 98 pages paper, which you easily can translate, this shows that the whole field of virology abandoned themselves. And the new field of virology only came into existence because Enders got the Nobel Prize in December for a speculation of the year, out of the year 48. And with the Nobel Prize, the other speculation that when cells are dying in the test tube, they transform themselves into viral matter, this became scientific fact through the Nobel Prize as well. And nobody was questioning this. And I mean, I have been long enough at university. When all of a sudden a colleague of yours or some professors publish a paper in Nature, really they treat them like God, and the Nobel Prize. I mean, that's no pope, no 100 popes together have such a wide, like a Nobel Prize in our society. And therefore, the speculation of Enders became a scientific fact. And a year afterwards, he wrote already, this paper of 54, this becomes the unique basis of measles virus production. Why? Because he lost, his opportunity for the polio vaccine. Jonas Salk was quicker. So, and you see it's all human what's happening. It's not falling out of the sky, and it's not by bad intention. Like I believed before, that they are all aware of those mishaps and misconceptions, and they would do it, even knowing that it's wrong. No, inside this field of cellular pathology, where only matter matters, you have no other explanation than decay or the devil itself replicating, virus, metastasis, and so on. So we are now, in order to understand this most important question, if there is a SARS-CoV-2 or other virus or not, we are in the point 4. So what they are doing, they put in statistical programs, they process those millions of molecules and then put them together in a process called alignment. And then they came up and said, look, this is the viral genome. And
no, this is such an interesting point, Stefan, because so many people, when you approach them with a paper that says, or people will approach me, or obviously not that I'm important, but Dr. Cowan and Dr. Kaufman and say, oh, here, this is the full genomic sequencing of this virus. And it's like, a genomic sequencing is not synonymous with the presence of an isolated virus, which is exactly what you're saying right here, correct?
Yes. So, but as a genome is the core of a virus, they claim if we fabricate the genome, it is there. But the belief system in which they are fooling themselves and the public is that when the cell's dying in the test tube, what they call the isolate, then it transforms the material, the animal material or the human material, it's transforming itself into viral matter. So, well, we take the smallest pieces, not wondering why we never ever got a long piece. Why not? So they are making the process of alignment and come up with a sequence, and they call it, this is the viral genome. And the one who is quickest, he is the name giver of his new idea, SARS-CoV-2, for example. And all the others, which are doing the same thing, of course, they say, look, I have a little bit, mine looks a little bit different. And so they call it, I found a mutation, right? Because they agree about 90% of the virus is not changing, but is especially on the gene for the spike protein, this is changing very much because that matter has a consciousness and it is changing. He's probably adapting better to the lung receptor and infecting more. And so the more successful would replicate quicker. Hey man, we would not survive a single moment if it's true like this. It's sure. Because when it's replicate bigger, boom, it goes like this. The next day it's over, right? So it's so anti-logic what they are doing, and we have a lot of examples. The swine flu in 2009, when the people became aware that there are very highly toxic nanoparticles in this vaccine, and the government and the army wouldn't get the so-called helping substances, the adjuvants, and they are free of nanoparticles, whereas the population should have it. So they refused, 93% of the population refused the vaccine, right? And then all of a sudden, the virus disappeared from every talk show, from one night to the next morning. It was over without vaccination. So the virus really behaved very well. He was so afraid against the vaccine, which has to be burned in the city of Magdeburg, you know. All the millions.
Such an intelligent virus. This virus is so intelligent.
And
right?
these contradictions, they are not seeing and they are not able to see. And Hammer, the one who really deeply understood and gave us back the meaning of life, and gave us back that everybody can check it out on itself, not being a specialist for atom theory or small genetic molecules adding to each other, alignment specialist, bioinformatics, you know, then you see that it's not possible in this way. So in this fourth point, they are not doing the control experiments to try to get the same alignment with equally treated cell cultures. Never, ever. You must imagine they do something to fulfill the model and forgot to check if they are not if it's not possible to get the same alignment result using exactly the same producer, but with sterilized so-called infected material. So then we go into step 5 of the—
Doctor, right before you get into that, just want to ask your opinion on this. So this guy, Anthony Fauci, that's going out there making public health recommendations. Do you, just your opinion, do you believe that he actually believes in what he's talking about?
Well, you should see that Fauci is more politician than a scientist. I can imagine that he has no idea what alignment meant, what they are doing nowadays about genetic things. No, he is a Cold War fighter, right? He became big in this system. And I mean, this is, I met Fauci in person, right? At this AIDS conference, International AIDS Conference in 98. And I told him in public at a press conference the truth into his face. And what happened, the press conference immediately was finished with my statement, right? No answer, no nothing. But it's impossible, or not impossible, but it's difficult. And this we have to admit. And Hammer found out that when something is touching us and what we feel, it's our identity. And he found the signal on the cortex would be around here on the left side of your cortex, and a certain organ is reacting also with this if your identity is touched. But most important,